Hello and welcome everyone to a special session responding to you, research on long COVID in Utah. My name is Ben Tomlin, Executive Director of Advancement Communications for the University of Utah. Now that we've been exiting this most pronounced past 2 years of the pandemic, the questions begin to surface: what does the future look like? And how does the disease affect those who have had it once the most obvious effects have gone? Today, we're going to take a look at a number of different issues and hear from a number of different panelists. First, I would like to introduce our host and real moderator for the day, Dr. Aaron Rothwell. Dr. Rothwell currently serves as the Interim Vice President for Research and Professor of Obstetrics and Gynecology in the Spencer Fox Eccles School of Medicine. She oversees 18 research administrative units that support research compliance, regulation and policies, and safety procedures across campus. In addition, Dr. Rothwell serves as the interim president of the University of Utah Research Foundation that cultivates research innovation and technology transfer for university real estate prospects and industry partnerships. Dr. Rothwell's research is currently funded through the National Institutes of Health and is focused on ethical, social, and legal implications of genetic and technological advancement on individuals and families, specifically within newborn screening, prenatal testing, and biobanking. I would now like to welcome onto the stage Dr. Rothwell.
Thank you, Ben. As Ben mentioned, my name is Aaron Rothwell, and I'm the Interim Vice President of Research here at the U. I'm honored to be here with these remarkable researchers and community partners as we talk about the U's response to COVID-19 and how we're moving forward on the long hauler research. The university has made extraordinary movements in these important areas of COVID-19, and the support we have received from our donors, our state leaders, our researchers, and community partners has contributed to these impactful discoveries. It's thanks to you and your support that I'm so excited to give you these promising updates today. Next slide. So first of all, what is long COVID? Today, when we think about long COVID, it is really what it is that when people get COVID, most of their symptoms can go away in a couple of weeks. However, as many as 1 in 3 people have long-term symptoms. This can include a wide range of health problems that consist for months or years. And personally, we have a colleague in the Vice President of Research Office that is currently struggling with long hauler symptoms. So we are seeing that every day. Next slide. COVID-19 research started at the U when we immediately recognized that this would be a once-in-a-lifetime public health crisis. Events seem to move so rapidly, it feels like it was just yesterday. It's hard to believe it was 2 years ago when we joined the national and international call to help fight this pandemic. The ability to mobilize investigators quickly is due to the University of Utah's robust infrastructure, our culture of collaboration, and the leading state-of-art facilities in the Intermountain West. This is what makes U so unique and ready to serve our public community. Next slide. So finding solutions, right? This is something that I'm very proud to share with everyone here today. So in addition to working with the Inflammation, Immunology, and Infectious Disease, which I will call the 3I initiative on, we were able to raise $1.3 million in COVID seed funding. Prior to COVID, we did have some systems in place to handle seed grant funding and to support researchers on novel studies. And that, it takes usually about 6 months to mobilize those efforts, to get the call out, to get, a group of panels, reviewers to review the applications. It took us 2 weeks. And I'm going to say that this was an amazing collaboration with 3I, and I am so grateful for Nicole Frank and all of her team for helping us address this important issue. And some of the early projects, they said, how can we detect COVID-19? How is COVID-19 impacting different groups? And what health disparities are we seeing? And what are the societal impacts of isolation and social distancing? It just wasn't the health that we were studying, it was also the social implications. Next slide. So when we think about how the $1.3 million was used to support our researchers, We currently have over 400 COVID-19 studies that are still going strong. We have $68 million in funding as a result of that seed funding, as well as some other special notable achievements, as you will see on this slide. But most importantly, the support from our donors and partners has led us to creating the U's Long Hauler Clinic. Next slide. Our work to understand long COVID started early in the pandemic when the Utah Long Hauler Group reacted to us to describe some of the challenges they were facing. Researchers moved quickly to explore these pressing needs. Then, with the support of a generous benefactor, we were able to streamline these projects to enhance the research infrastructure. All the while, we created a clinic that serves patients and enables researchers to partner with patients in real time to understand their symptoms and treatment trajectory. To date, this clinic has served over 600 patients with specialty care across 35 different areas. In our first update in June in 2021, we shared that long COVID was going to become a primary research area for investigators. Today, you will hear about the work that they have been up to. And I'm going to pause here because I think we have a pleasure of sharing a very short video that illustrates the breadth and strength of our COVID Research 19 here at the U over the last 15 months.
Before COVID, I went on a bike tour in France. And so I was in really good shape physically. And I was walking a lot, working a lot, just living my life, playing music. I was one of the first 20 cases in the state. So I got it in early March. I was so just beat down by it. I couldn't get out of bed. I couldn't go up the stairs. And I didn't have the aching in my body until maybe 3 months after. And then I had severe pain. So I'm well into over a year. And my doctor then said, there's a long haul clinic.
So we opened in mid-July of last year, and we have seen about 600 patients so far. We have provided referrals to 35 different specialties. The most common specialties being sleep medicine, pulmonary medicine, cardiology, neurology, and social work.
I kept going like, oh, well, I'm not making this up. I don't think I'm making this up, but I'm not as sick as everybody else. I didn't go on a respirator, or, I can still do things. So, and they just right away said, no, you're not making it up. This is real.
It's a whole spectrum of symptoms. We're seeing persistent fatigue, shortness of breath, palpitations or heart racing sensations, lightheadedness, even loss of consciousness. A lot of the typical symptoms that we see in patients with cardiovascular disease, there's a lot of overlap in patients with this post-COVID syndrome.
Patients, one of the things they ask for is what's going on with research, what are we learning, how can I help?
What makes the University of Utah well suited to do research on COVID and long COVID is really vastly intertwined with what makes the university an excellent research institution. We have the breadth of clinical and bench or wet lab researchers. We also have an extremely good epidemiology group. The work that we have just started doing here is a study called Recover. We've begun collaborating with 15 adult sites, 6 or 7 pediatric sites, and 2 pregnancy sites around the country. That study is enrolling people who have or have not had COVID and either do or don't have long COVID symptoms to understand what the disease course might have been that led them to long COVID, whether or not there's a genetic predisposition, what blood tests like your white count or your course of disease early in your COVID course may predispose you to long COVID, whether or not you've been vaccinated or not vaccinated, and then trying to understand what the different complications are of long COVID and if we can begin to understand the mechanisms behind that.
We've been working with the Long Haulers Clinic in terms of a study that we're doing where we're following people who had COVID during pregnancy. to see if they develop long COVID. And we're also following their babies to see if there's any adverse effects on the children from being exposed to SARS-CoV-2 while they were in utero. What we found is that people who had SARS-CoV-2 in pregnancy were at higher risk for developing really serious pregnancy complications, as well as for death during the pregnancy. That also translated to risks to the neonates as well. We saw increased risk of preterm birth, increased risk of neonatal ICU admission. And so really saw increased risk for both mothers and babies who had SARS-CoV-2 in pregnancy with the delivery itself and with outcomes surrounding the time of delivery itself, which certainly could also translate into longer term consequences for both the mother and the baby.
Dr. Adam Spivak and I, he's from infectious disease, we are co-principal investigators on a small biobank here at the University of Utah, which is attached to a self-enroll registry, meaning these are patients that are seen in the clinic and patients that we don't see in the clinic that can enroll in a registry, provide their demographic information, provide what symptoms they're still having after their COVID-19 illness. And with that, there's an option for them to provide their blood to have it biobanked, spun down and frozen. And the idea there would be over time, we can test their blood and try to identify patterns that may help us understand what's actually causing this post-COVID syndrome.
I think one of the challenges as physicians sometimes is that if there's not a broad aspect of research to go to base things on, it can feel like you're a little bit sort of making it up as you go. And it can be hard to tell patients, I don't know. But partnering, I think, and listening to people and really understanding what their concerns are and being able to say, I don't know exactly what's going on, but we're going to partner through this and help each other out as best we can, I think has been a positive message for patients.
One of the coolest parts of this whole story for me is to see a community reach out to a university and say, hey, we've got a problem, and to not just throw their voices at it, but even throw their money at it and say, let's try to make this better. That's been a very unique part of this journey.
I think the long COVID clinic is frankly amazing, and they're so reassuring that no, you're not making it up. We have lots of patients like this. I can't tell you what it means to me to know that there's a place I can go, and even if they don't have all the answers, they're going to listen, they're going to problem solve, they're going to help. It gives me hope. If I have another relapse or something, I'll go back up there, and if there's something they can do, they'll do it.
All right, that was a very inspirational video. As a university that is for Utah, we are proud to have partnered with our community to address this growing need, both clinically and through research. There's a unique synergy at the U because we are a leading academic medical institution, and we have the valuable opportunity to partner with our patients and community members to learn how to better take care of Utah. Next, you will hear from some researchers themselves who are leading these pioneering efforts. There will be time at the end for questions, so feel free to write your questions in the chat, or you can speak up at the end. So the first person I would like to introduce today, we have Beth Williams. Ms. Williams is a 6-year PhD candidate in the Pinelli's lab. Ms. Williams is investigating what happens to the body and in the immune system to develop long COVID. Ms. Williams, I'll turn it over to you.
Thank you, Erin, for that introduction. Okay. Thank you, everybody, for your attention. And I'd also like to thank the III donors for making my research and a lot of other COVID research here possible. Okay. Just a brief timeline. At the end of December 2019, the World Health Organization was notified of a pneumonia cluster in China. And by the middle of March, the WHO had declared this a pandemic. Amazingly, by the end of 2020, there were 2 vaccines that were recommended for everybody over the age of 16 to help prevent severe COVID disease. But still, as of the middle of April, there have been over 5 million cases of COVID-19, as well as approximately 6 million deaths. And amazingly, over 11 billion vaccine dosages administered. But what we're talking about today is long COVID. And long COVID affects 10 to 30% of individuals who were infected by SARS-CoV-2. So what is long COVID? People who had COVID continue to have or develop new symptoms that impact their everyday life. And they seem to occur or continue occurring up to and through 3 months after initial symptom onset. The main symptoms are fatigue, joint pain, chest pain, shortness of breath, persistent cough. And these are the 25 most common symptoms, but there have been over 250 symptoms of long COVID described. We don't really know what's causing this. The Planeus lab has been studying virus-host interactions at the University of Utah for over 17 years in the context of HIV. And in early 2020, we pivoted to studying neutralizing antibodies to SARS-CoV-2. Our lab, as well as in collaboration with ARUP and other III investigators, were quickly able to contribute to the world scientific stage trying to unravel this major world conundrum. Additionally, one of my mentors is Adam Spivak, and he was integral in starting the long COVID registry and biobank, and I have access to all of those samples for my scientific analysis. Additionally, the link that is showing on the screen will be copied and pasted into the chat either now or when I'm done presenting. Individuals who sign up are asked a detailed symptom survey and then asked to donate their blood. So to study long COVID, we've been taking samples from the biobank and comparing them with samples that I have collected in my lab. And these samples come from similar demographic profiles, except that all of the people who have donated their blood for long COVID research are all female. So what we test are called cytokines, and they are in your blood. Cytokines can be called cell messengers or hormone-like or similar to a cell's Wi-Fi signal, and they are generated by cells and come out of cells and then are picked back up by either the same cell that created it in order to cause a response. And in this little cartoon, the cell is turning yellow with a pink membrane. But this cytokines can also be picked up by other cells and generate a response in a much wider fashion. So we sent these samples to our collaborators at ARUP to measure 13 different kinds of cytokines, and they can all either increase cellular or body inflammation, or decrease inflammation, or induce wound healing. On this graph, on the left, we're going to look at the amount of cytokines, and on the bottom, we're going to look at how the amount of cytokines change over time. So what happens when somebody gets sick, they come in contact with the virus, and an amount of cytokines go up, and then it drops, and it returns back to baseline. So we hypothesized that with long COVID, what was happening was that cytokines were going up, but then were never returning back to normal, and that the sustained high level of inflammatory markers in your blood was just wreaking havoc everywhere in your body. But what we saw, in fact, was surprising, because the level of cytokines that we found in the blood of people with long COVID were way below baseline and even at zero. I'm going to try and put this into context of the immune system. Here are 10 of the most common immune cells, and we're going to focus on just a few of them. IL-6 is a cytokine that is secreted by these 3 myeloid lineage cells on the left-hand side of the screen, and they affect T cells. Once activated, T cells do many jobs in the immune system, including secreting IL-2, which activates other cells. And if you look at this graph, the colors will be repeated throughout the slides. The individuals with long COVID are in blue, and the individuals who are healthy are in green. And you'll notice that IL-6 and IL-2 are both lower in people with long COVID. So without T cells, there is no sustained activation of T cells or B cells, which means that there are no cells specifically to target cells that get infected with a virus, which means that even if you've cleared SARS-CoV-2, you might not be able to fight off a new infection, which could cause your body to feel ill as you slowly fight off something that you may otherwise would have been able to clear very quickly. IL-6 again is secreted by T cells, and once activated, T cells secrete IL-4. IL-4 is responsible for B cells, which produce antibodies. And antibodies are something we've all been learning a lot about in the last couple of years, because the vaccines induce antibodies, and the antibodies are what help protect us from getting severe infection if we get infected after receiving the vaccine. And without these, again, there are no antibodies, no cells specifically to target infected cells, making it very hard to fight off new infections. IL-8, and you can see that there is no blue bar, causes neutrophils to traffic to the site of cellular damage. And in COVID, that's the lung system. So if there are no neutrophils going to your lungs, they're not going to heal properly. And these may be the cause of prolonged cough. Interferon gamma is a major effector of the immune system, and it works to activate many of the cells in the immune system, as well as IL-17. And you can see all of these cells have gotten bigger. These two cytokines work to help clear pathogens from your blood and return your body to normal. And you can notice the blue bar is much smaller than the green bar in both of these cases. Lastly, IL-13, again, made by T cells. So if we can't activate them in the first place, we're not going to have any of these last three cytokines. IL-13 is responsible for putting the brakes on the immune system. And without it, we're going to have overactivation, a lack of healing, and no return to baseline. You're beginning to see how things are dysregular and not working normally. So in summary, we're seeing a state of immune exhaustion where we have less production of cell messengers, causing less response from cells, creating a negative feedback loop. We are working to figure out how to solve this problem. And to do this, we have new samples that have been donated at the biobank since I went and got them a couple months ago. And we also have access to samples from Dr. Spivak's hydroxychloroquine clinical trial. And these are pretty amazing because there are 3 samples or 4 samples per individual, right from the beginning of infection, mid-infection, late infection, and then 6 months after. We hope to be able to get a really great snapshot of what's happening over the whole course of recovery. And so far, there are 82 members that have completed all 4 blood draws, which means that there are approximately 27 of them who likely have long COVID. We'd really like to do some high-powered single-cell RNA sequencing where we can see what's happening in every individual cell, as well as screening to identify pharmacological intervention that will help initiate restoration of immune function and provide some relief from the suffering of long COVID symptoms. Cytokines are really just the tip of the iceberg, and we're hoping to do the single-cell sequencing because we'll be able to get a huge understanding of what's going on when we can look at how 25,000 genes per cell are all working together to cause the systemic issues that we're seeing. So again, we see a state of immune exhaustion, and we're really hoping that with continued research, we'll be able to come up with a therapeutic option for this. And with that, I would like to thank my advisor, Vicente Planeas, my mentors, my collaborators at ARUP, the individuals with the Long COVID Registry and the Biobank, and all of the people who have donated. If you know anybody who has long COVID or have long COVID yourself, we would really love to have more samples to analyze. So please come and donate. Thank you.
That is fantastic, Beth. Thank you so much. These are promising findings to explain how the immune system is malfunctioning in long COVID. I should also highlight that a generous benefactor who invested in trainees in this research space, Ms. William, is also a 3I initiative, a 3I fellow in emerging microbial threats. That's a mouthful. This fellowship ensures that she has time to dive into these important questions. And again, I'm starting to see some questions in the chat. We will get to those as they emerge. Please put those in the chat. Now I'd like to introduce Dr. Molly Cummings, who will share about how long COVID is seen across the populations in Utah. Dr. Cummings is a biomedical informatician within the College of Nursing and the School of Medicine. So I'll give it to you, Dr. Cummings.
Thank you, Erin, for the kind introduction. So, as you heard from Beth Williams, many people who have been ill with COVID-19 are struggling with lingering symptoms or even new health problems long after their initial recovery from illness. And we often hear this referred to as long COVID. More formally, we call it PASC, post-acute sequela of SARS-CoV-2 infection. The symptoms can be mild or quite severe and incapacitating. But how many people are suffering from PASC and suffering from apparently different types of PASC symptoms? Why do some people get PASC and not others? What causes it? We don't yet have concrete answers to these questions, but it's extremely important that we find answers, and soon. Given the number of people infected with SARS-CoV-2 in the US and the world, PASC has very important implications for many people's lives and well-being. And these individuals will need health care and treatment into the future. So in order to help individuals suffering from PASC and to make good decisions in healthcare policy and planning, we need to know more, and we need to know more as quickly as possible. So in 2021, the NIH announced a new initiative focused on supporting research related to the prolonged health consequences of SARS-CoV-2 infection. This large-scale initiative to understand, prevent, and treat PASC is known as RECOVER. Dr. Rachel Hess talked about it briefly in the introduction video. It consists of multiple large-scale studies and projects, each with a specific focus, including the study I'll talk about today. The study I'm describing today is called RECOVER EHR, Electronic Health Record. And in this RECOVER study, we are working together with other health systems and partners across the country, pulling our electronic health record data together in a secure enclave so that we can use it to ask a wide variety of questions related to COVID, using traditional statistical approaches, but also methods of artificial intelligence and machine learning that are particularly well suited for finding patterns in this type of data. Our goal is to create definitions of long COVID that can be used to support research studies. We'll be able to look at subtypes of long COVID and specific symptom clusters. We'll study risk, mitigating and protective factors across many different people and communities. We'll build models that predict outcomes based on those factors. We'll study racial, ethnic, and socioeconomic disparities of long COVID, and we'll analyze the utilization of healthcare resources. These are fundamental types of information that we need to characterize and further study long COVID and to plan for the future care of affected individuals. In doing so, we take advantage of the benefits of using real-world data, the data routinely collected in EHRs. This type of data, millions of records from across the country, is advantageous in many ways. Its sheer scale gives us the capability to analyze the data in highly flexible ways, look at change over time, geographical variation. With this much data, you can effectively examine recent trends or changes and carry out rapid initial analyses that can be followed by more formal studies. Recover EHR is building upon the Patient-Centered Clinical Research Network, or PCORNet, for data science research related to COVID. This is a nationwide clinical research network, and University of Utah has been a member organization of PCORNet since 2015 in a large effort led by Dr. Rachel Hess. PCORNet has made a number of large-scale patient-centered outcome studies possible. Through participation in PCORNet, University of Utah researchers can access data from other sites across the country for research. This helps them study rare diseases and subgroups of people in ways that wouldn't be possible at a single geographic location. In building PCORNet, we established the infrastructure for sharing EHR data using a common data model. You see, the data from all these different sites is very different in how it's structured, whether it's collected at all, and how often it is collected. This makes pooling the data for research a very complicated matter. And if we had to start from scratch for Recover EHR, it would take years to accomplish. However, PCORNet has an established common data model, a common language, if you will. We already have systems and processes for translating our EHR data to this common language and pulling it together across sites. Recover EHR is building upon this existing capability so that we can move quickly to our scientific goals. When we want to study patients suffering from PASC, one of the first questions we ask is, who has it? Who has PASC and who doesn't? And it seems like a simple question, but with electronic health record data, it requires the use of what we call a computable phenotype. Here you can see an example of how we might create a rule to find patients with diabetes. This is a definition that when coded, a computer can understand. And so we could use the code to answer the question, who has diabetes, in a large group of electronic health records. However, when a disease is new, the rules are not clear, and we need to use more advanced and creative approaches. Fortunately, as long as you have a large group of patients who you know are suffering from this new disease, you can use methods of artificial intelligence and machine learning. This is true for PASC and its subtypes. Here's a visual. We have some patients who we can say with confidence are suffering from PASC or some specific subtype of PASC. And those are the patients in the blue rectangle, the blue bubble. And for Recover, we're using patients who have been diagnosed with PASC in long COVID specialty clinics. Using artificial intelligence and machine learning for pattern recognition, we then find patients who are similar to them in terms of their symptoms and other patterns in their healthcare data. And so we can say with confidence that these other folks, although they were not seen in a COVID specialty clinic, but in all likelihood, they also have PASC. We can do this for PASC in general. We can also do this for various subtypes of PASC. And once we can name it, once we can label the data as examples of PASC subtypes, or none at all, we can start asking and answering a wide variety of questions and identifying groups of records for more specific studies. During the current year of Recover EHR, our first year, we're focused on this scientific work. First and foremost, detection, the ability to identify and characterize PASC and its subtypes. Then we delve more deeply into risk factors, disparities. We'll look at different treatments and how well they seem to be working, and learn whether vaccines are effective in preventing PASC and its subtypes. Why is it important that we participate in this study? Why Utah? Why make sure our data is included in any of the Recover studies? Well, it's important that the research findings at a national level reflect Utahns with their unique characteristics and health concerns. In general, people living in rural and frontier areas like those that we serve in Utah are underrepresented in health research. And the fact is, our population has fundamentally different characteristics and considerations than populations in other parts of the United States. We have distinctive regional considerations, such as air quality. We know that air quality affects pulmonary symptoms, breathing symptoms, which are important for long COVID. Essentially, if we want the research findings to be relevant for Utah, we need to make certain we're part of national research efforts. Also, this type of nationwide teamwork enables rapid discovery. We can work faster to find conclusive answers to many of our questions about long COVID. We leverage a much larger team of scientists and data experts to find good answers fast. And finally, we're creating an important national data resource that our Utah-based scientists will be able to access into the future for research they would like to initiate and conduct. In conclusion, the University of Utah is engaged in a large-scale national effort using the power of data and teamwork to advance the detection, prediction, treatment, and prevention of PASC. The project is expected to generate a great deal of insight into the extent and nature of PASC, as well as provide critical data tools to support the RECOVER cohort studies, which will use outcomes from this project, but also enroll individual patients suffering from PASC here in Utah and across the country. I'd like to acknowledge our very dedicated and talented interdisciplinary team here at University of Utah, with faculty and staff across nursing, biomedical informatics, pulmonology, population sciences, and our UHealth Enterprise Data Warehouse team. And if you'd like to learn more about RECOVER, I encourage you to visit this website or reach out to me directly. I'd be happy to discuss the project. Thank you for your attention.
Wow, that was so interesting, Dr. Cummings. It's fascinating how quickly you're trying to define what COVID is, so you can look for how it impacts people across our country. So thank you. All right, we have our next speaker. I would like to introduce Dr. Tori Metz, who you heard from briefly in the video. Yeah, she's also an actress. We will describe how long COVID affects special population, people who are pregnant and their babies. Dr. Metz is the Vice Chair of Research and an Associate Professor in OBGYN at the University of Utah Health. She is also a practicing maternal fetal medicine subspecialist. So again, welcome, Dr. Metz.
Great, thanks so much for having me. Just wait for my slides to come up. Excellent, thank you. So thank you all for joining, and I'm happy to be here today to speak with you a little bit more about COVID-19 and pregnancy. And I heard a little bit about it in the video, but I want to elaborate on some of those points. So, obviously the pandemic has affected everyone, but pregnant people have a particularly unique risk of COVID-19 and pregnancy. And more specifically, they have a greater risk of severe illness. There was a CDC MMWR report that came out early in the pandemic that really drew our attention to this, of almost 500,000 reproductive age females. And what they saw is that people who are pregnant have an increased risk of ICU admission, need for intubation or mechanical ventilation, and death with COVID-19 and pregnancy compared to those who are not pregnant. So when we think about obstetrics questions and questions about pregnancy in general, there's two things we usually think about. One is, how does a disease affect pregnancy? And then how does pregnancy affect a disease? And so those were the types of questions that we asked early in the pandemic. How does COVID-19 affect outcomes for mothers and babies? And how does being pregnant affect the disease course of COVID-19? I was really motivated to plunge into this work. I'm a physician scientist, as Dr. Rothwell mentioned, so I am a practicing maternal fetal medicine subspecialist. And so I was seeing patients who had COVID in pregnancy, and we were really struggling with what to tell them because we just didn't know. I was also the director of labor and delivery at that time, and so I was having to create policies to really try to keep our patients and our staff safe early in the pandemic when we still didn't even know how SARS-CoV-2 was transmitted. And at that time, I noted, we have access to this large obstetric clinical trials research network that University of Utah has been part of now for over 30 years. And this is a real opportunity for us to contribute information to patients in the community. So we started what is called the GRAVID study. This was done through the National Institutes of Child Health and Human Development and the Maternal Fetal Medicine Units Network. We call it the GRAVID study, which is Gestational Research Assessments of COVID-19. And this network involves 12 centers. Usually these centers do obstetric clinical trials and really help shape the way that we practice obstetrics in the United States. But in this case, we really wanted to put together an observational study that was comprehensive across the United States. And so at those 12 centers, we included 17 individual hospital sites. And you can see Utah there is really that lone ranger out in the West. So really important for us to have a voice at this table and make sure that the population is more representative. So what we did was a retrospective cohort study of 17 U.S. hospitals who are participating in this network. And in total, we included 14,104 pregnant or postpartum patients who delivered from March through December of 2020. 2,352 of those patients had SARS-CoV-2 infection, and we compared those with those without SARS-CoV-2 who delivered on randomly selected dates, and that accounted for the other 11,752 patients that were included. And we had perinatal research staff that really went through and abstracted these medical records in detail to make sure that we really understood the COVID-19 course for these patients, as well as their pregnancy outcomes. And what we found is that people who are at risk, who had SARS-CoV-2, were at risk for pregnancy complications. In terms of neonatal complications, we saw that people who had SARS-CoV-2 were at higher risk for preterm birth and admission to the NICU, that their babies were at risk. And we also saw maternal complications. So what we looked at was death and then complications from things that we see pretty commonly in pregnancy. So we see people who develop high blood pressure, bleeding after delivery, or additional infections associated with the time of delivery, but really looked at complications from those things. So not just having high blood pressure, but having a stroke because of high blood pressure, or requiring acute IV administration of medications to try to prevent that from happening, or having seizures from high blood pressure, or damage to the kidneys or the liver. Same thing with the bleeding. We weren't just looking at any bleeding that occurred after delivery, but really people who needed higher interventions for bleeding. So things like surgical procedures to control the bleeding. And with infection, we really looked at people who needed their wounds reopened after C-sections or really deep infections in the abdomen, not just having an additional infection, but really being sick from these complications. And people at SARS-CoV-2 were at higher risk for these things. So what about long COVID, which is really what we're here to discuss today? What are we doing about that? What are our next steps? Well, the reason I shared with you that initial data was really because that was really the basis for us being able to do more research here at the University of Utah to now examine what happens to these mothers and babies after they have SARS-CoV-2 in pregnancy. So as part of this recovery initiative, we're doing what we're calling the PASC-PREG study, which is a 4-year observational study. We're enrolling individuals with SARS-CoV-2 in pregnancy and their offspring. And our target enrollment across all of these maternal fetal medicine unit network sites is 1,500 dyads, so 1,500 mom-baby pairs. And we're looking at outcomes in both the mothers and the babies. So in the mothers, we're looking at the long-term sequelae, like we've talked about in the other lectures today, across organ systems. And again, this is just a smattering of what's out there. We know there are so many other symptoms that are associated with long COVID. We're looking at things like neurologic and cognitive decline. From a psychiatric standpoint, we're looking at depression and anxiety, especially peripartum occurrence of these events. Cardiac disease, arrhythmias, cardiomyopathies, hypertension, POTS. From a respiratory standpoint, shortness of breath, chronic cough, the development of diabetes. And then from a reproductive standpoint, there's a lot of interest in changes in menstrual cycles and fertility. And that's something that we're looking at in this population as well. So how is pregnancy different? Why is this separate? Why are we looking at this cohort specifically and trying to enroll 1,500 people who had SARS-CoV-2 in pregnancy? Well, the immune system is just more tolerant in pregnancy. You can imagine that it needs to be, right? You have a fetus that a mother is tolerating, and that fetus has DNA. Half of the DNA is totally different from the patient's partner. And so it's really remarkable that mothers are able to tolerate that at all. And so really unique things happen to the immune system during pregnancy. That's also partly why pregnant people tend to get sicker with viruses. And that's not just SARS-CoV-2, that's flu, that's any viruses. We really see pregnant people tend to get a little sicker, and that's because they have more immune tolerance of these foreign things. We also see, we wonder if we'll see that decreased inflammation of getting SARS-CoV-2 during pregnancy may result in less PASC, or alternatively, these patients are getting more severe disease and infection, so maybe that will cause more PASC. We don't know, and this is one of the things that we're studying with the Recover Initiative. And the other question we're asking with these dyads is, how are the offspring affected? When we have SARS-CoV-2 exposure in utero, we know that there's maternal hypoxia and there's or low oxygen saturation, and there's an inflammatory response. And that results in placental damage, which is important, right? Because that's a connection between the mother and the baby to provide nutrients and oxygenation. That damage can result in adverse outcomes like stillbirth, preterm delivery, and early onset preeclampsia or high blood pressure. And we're worried that that damage could also result in adverse long-term outcomes. So what we're looking at in Recover is delays in social, emotional, and neurobehavioral development between those who are exposed and unexposed to SARS-CoV-2, and whether there's adverse cardiometabolic outcomes for those offspring in the long term. Are they predisposed to obesity and other cardiovascular adverse events? So in conclusion, I want to just say that pregnant people are at high risk with COVID-19. We're concerned that SARS-CoV-2 may affect mothers and offspring long term, and that's what we want to study now. There's really a need for ongoing high quality work in this population. And so we're really fortunate that University of Utah is part of this Recover Initiative. I definitely want to thank the National Institute of Child Health and Human Development for funding the initial study that laid the groundwork for all of this, and now the National Heart, Lung, and Blood Institute for funding the Recover study that we're part of and enrolling actively in now. And all of you for joining today. Thanks so much.
Wow, that is impressive, Dr. Metz. And also it's impressive to see how quickly you were able to leverage this research network to learn more about how COVID impacts pregnant people and their babies. Really important. And I just want to also echo that I want to thank all of the speakers. You can see that investigators here at the U are approaching long COVID from many angles and trying to get a clear picture of what we are encountering. So thank you again for all your time today. So how can you help? Let me see if I can move my slide myself. We also, this is a joint effort, right? We invite you all to be part of this effort. And I think the research, as well as, the satisfaction from being a scientist, I think it benefits from these strong collaborations with all of our communities, donors, and other researchers. So if you'd like to get involved, there are 2 study opportunities you may want to consider. You heard one today about the exciting discoveries with long COVID, which was made possible by the Long COVID Biobank. If you've had and you want to participate, here's the link there. If you had COVID or still didn't have COVID, but you would like to participate in the NIH recovery study, you can see here what is required and the website where you could email to ask questions. And of course, all of this research requires a great deal of financial support, and we'd be incredibly grateful for your partnership to continue to pursue this important research. The U is currently working on many critical projects in medicine and healthcare, as well as societal challenges on how COVID impacts mental health, social injustices, racial disparities, economic recovery, access to education for K through 12, and so much more. If you are interested in providing any type of seed funding for any of these areas of research, please consider donating at givetoutah@uuhealth.org. Many of you have seen today with our speakers and videos how small amounts of money and little moves can result in big returns. Your support truly makes a difference in research and recovery. So again, thank you to all of our partners as we continue to support research on long COVID. We are truly grateful for your commitment and just the opportunity to collaborate with all of you. It really makes everything a lot more rewarding. So thank you. And now I'm going to turn it over back to Ben, and me and him are going to QA some moderation.
Thank you so much, Dr. Rothwell, and to all of the panelists. Actually, I am going to invite all of our presenters including Dr. Rothwell, please to come back on stage if you would. A number of questions have been saved up in the chat, and we're going to get to as many of them as we can in the remaining 10 minutes or so. Thank you to everyone who has put in. Some of them I've noticed that the panelists have replied already in the chat, and so hopefully you've gotten some answers there. One of the first questions I'd love to set the stage with, though, that I'm very curious about is this unusual for a disease? Is COVID unusual in the long-term trailing effects that it has? On the one hand, we started to hear at the beginning of the pandemic that it's a coronavirus, right? That coronaviruses are of a family of virus that can be fairly common. And yet the way that it seems to have attacked our system, and now the length, the long tail, seems to be unusual. Or is some of this also an effect of this is the first global, really truly global pandemic of this size to happen with modern medicine and all the tools that we have to study it in blisteringly real time. So I'm curious to know how much of it is, are we meeting the moment to get to know it better in real time? Or is it truly a unique disease?
I can take that question. It is not unique in causing post-viral syndromes. The initial SARS outbreak in 2003 had a post-viral syndrome with very similar symptoms. And MERS in 2012, I believe, also has a post-viral syndrome. And it tends to have a few more physical or a few less physical symptoms, but more mental health of impact rather than the symptoms we're seeing physically with SARS-CoV-2. Chikungunya also has a post-viral syndrome that causes rheumatoid arthritis-like symptoms. And there's even been literature that suggests that extended periods of time in the ICU, no matter what sort of respiratory disease you have, causes a syndrome that they're calling post-ICU syndrome, with depression and anxiety and just physical malaise that is actually quite common.
I remember in talking with some of the folks, some patients here at a previous event last fall that had mentioned that they've also been met a little bit with stigma or in cases of some of their healthcare providers, a disbelief that the symptoms are continuing. I'm wondering if anyone could speak to that. Is some of this also that we need to educate the medical community as we're all getting up to speed? I'm not sure who might be the favorite to take that, so I'll throw it out to the whole group.
Sorry, I pushed unmute. I can take that as a physician in the group. So, I mean, I do think that, you know, obviously this is a real thing, and I think that that's one of the things that was stressed in our video about long COVID. And I think that that's what's been so important to patients, about being heard about their symptoms and really feeling like somebody's looking into this. And I think that's one of the really wonderful things about the long haul COVID clinic that we have here at the University of Utah, is that, you know, that validation that these are real symptoms they're having. And then, you know, accompanying that with this research component where we're trying to look at why and, you know, what's different between people who develop PASC and don't develop PASC. And then obviously the next step is intervention. And that's something that's already being discussed in all of these research networks is, you know, if we know what predisposes people to long haul COVID, then how can we prevent it? Because obviously that's what we all want to aim towards. And if in people who are already experiencing it, how can we help make it better? And so I think that, you know, initially, as we start to gain more information about this, a lot of that information at first is just why, who, how, what's happening. And then the next steps, the next natural steps that people are already thinking about and working on is then how do we treat this? How do we make it better? How do we prevent it in the first place? And I think that's where we're moving, but we can only move that way with getting the word out and validating. And I do think that, you know, people do recognize this here, and there's a lot of messaging that goes out to the doctors here about, we have this long haul COVID clinic, let's get people into it. And so I think people do understand that this is real and that we do need to get patients help.
Laura asks, can people get long hauler COVID if they've been vaccinated and boosted? And then how could they test there? Or how might doctors test to prove that that's causing the issue? I think. Sorry.
Yeah, I mean, I think that's another question that we definitely want to answer. You know, is there risk reduction if people get COVID after they've been vaccinated? There are a lot of things out there that would suggest yes. I mean, if you extend the work that's been done and, you know, pediatrics that have demonstrated an increased risk of, you know, multi-system inflammatory disease in kids who have been vaccinated prior to contracting COVID. So we do see that. You know, a lot of the hypotheses surrounding these longer term effects are related to that inflammatory effect and higher disease severity. And so if we can prevent some of that with vaccination first, I mean, I think pathophysiologically, it makes sense that people are going to have a lower rate of these symptoms if they've been vaccinated, but doesn't mean it never happens. We've certainly seen it. You know, there are people who have been vaccinated who have then gotten COVID, who then still have long haul symptoms. So it happens. How much, you know, that risk is reduced by being vaccinated in advance is one of the questions that we need to answer. And I think one of the questions is going to be possible to answer with some of these big data projects like the PCORNet project.
And in fact, some of the preliminary data that's been shared in our steering committee meetings is consistent with what you just described, that in pediatric populations, they're seeing that you are more likely to have long COVID if you were not vaccinated. But that there should be some publications coming out of the PCORNet Recover EHR project within the next few months with a more detailed analysis.
And Molly, you actually have another question here about how the EHR data, is it matched with other data that may live outside the EHR record, such as income, education, self-reported stress, or community factors to build that model?
That's a great question. In this first year, we're linking it with vaccine data and also death data, mortality data. We are able to get at some data describing social and environmental determinants of health as it's linked to geography. But to protect the anonymity of the patients who are described in this data, we can't share precisely detailed geographic information. We can loosen up the geographical area with which an individual anonymous patient record is associated. And then we can look at that geographical area and the characteristics of that community. And that allows us to have some insight.
There's another question here that I think I'm going to read it live because I think this is a knowledgeable person asking the question, but PASC systems are highly reminiscent of those associated with chronic fatigue syndrome and fibromyalgia. Are there any biomarkers that cross over among these? Might the COVID-19 research provide insight into these and other dysmodulation syndromes?
I can answer this, and I don't know if Tori will have anything to add, but there are a few published fibromyalgia papers that I was looking at to compare my cytokine results directly with. And in many of the people who suffer from more severe symptoms from fibromyalgia, the cytokines that I looked at were also significantly lower. And I am hoping, I'm trying to write an IRB now to study that in parallel and see if we can bring a little bit more to light.
We're nearing the end of our time, but I want to see if we can just get two more questions maybe in here, if everyone would like to stick around for just a moment. I think another question here was women's health, and are we looking at it beyond just pregnancy? Lisa asked the question that there's other evidence of effects of other portions of women's health.
Yes, absolutely. That's definitely something that we're looking at in the Recover study for adults overall. We know that PASC seems to disproportionately affect women. And so it's definitely something that has come up. I know talking to Dr. Brown, who is the medical director of the long COVID clinic and was in the video, we've developed now a pathway for patients to be referred who have abnormalities in their menstrual cycles after COVID-19 because that's definitely something that they're seeing. And we are developing now ways to sort of have patients track that, begin to gather better data about that. But we're also looking at it in the RECOVER study because it's an incredibly important aspect of, women's health.
This might be a question then for you, Dr. Rothwell. It's obviously incredibly impressive how we're bringing together all of these elements, this data research, the biomedical research, the clinical care. From your standpoint, sort of seeing all of these integrate together, what are the advantages that we get, I guess, by having all these boats in the water at the same time?
Well, I think, I talked about it earlier. I think it's the quickness of the response, moving the review process from 6 months to 2 weeks, right? I think it's the nimbleness. So I think we're now in a position, so if there is an emerging hypothesis or something that, let's say Dr. Cummings or Dr. Metz is seeing, I think we're well positioned to go, well, let's move on this, right? Versus waiting 3 months to submit the application and get it going. Something I also think is very important to mention is that, I was running the IRB at that time, and we prioritized all COVID studies first, right? I mean, and so to continue that prioritization for important public health topics, I think will help drive our efficiency. So I think also I want to thank the IRB for their hard work and all this research.
Finally, one last question, then I'll just open up for any closing thoughts that anybody would like to share. Kathy has shared that she's on the line, that she's having her own experience, I think, with the long hauler syndrome for as much as 15 months. And it seems to be affecting her abilities to breathe and throat. Now, obviously, I think we have talked to it, but I'm just wondering if we have any thoughts for her or anybody else who's on the line who feels like they may be having these experiences. Not the least of which perhaps is a recommendation to visit the clinic or come see us.
Yeah, I think definitely to get, into the long hauler clinic, and then also, potentially would be a candidate for signing up for the RECOVER work that's being done in the adult cohort here. I talked about the pregnancy cohort, but Dr. Hess is leading an adult cohort where you don't have to have SARS-CoV-2 in pregnancy, just any time in adulthood. And you can email Utah Recover at hsc.utah.edu to get involved with that work.
That's great. Any closing thoughts from anybody? Topics that we might not have gotten to, or points in the questions that you might see here?
I would just like to continue to support this research. I think this is highly important. And I think also maybe highlighting, more of the mental health and the social injustices and effects of isolation. I think that would be very exciting to see how we can develop interventions to help people better recover this, even outside the clinic.
Well, terrific. Well, we'd like to, first of all, thank you very much for all the work that you've been doing. I think on behalf of all of us at University of Utah Health and the university who have been watching how people have responded over the past 2 years and the enormous amount of work that gets put in, it is both inspiring. We know that at times it can be stressful, but at the same time, everybody who seems to be doing this work has demonstrated a tremendous amount of passion. And the response and effect has been remarkable to watch. So thank you for this work. Thank you for everyone who has attended for today. A number of links have been shared in the chat, including how to participate in the studies, how to make donations, or how to support the work in other ways. And we hope that and encourage you to follow up with us. This session will be recorded, and so you can go back and watch it later. And please share with others as well. Dr. Rothwell, I'll just turn it over to you one last time in case you have any closing remarks or thanks. And otherwise, I would like to thank everybody for their time today.
I'd like to thank, obviously, the researchers, but also III for their continued infrastructure and support for keeping this research going. So thank you to everyone.
Thank you all. Have a terrific day.